All right, everyone. So it's almost 1:10. So we start making starts. Welcome to this afternoon's same plenary, which is going to be given by Professor Giulia Giordano. So Giulia is from the University of Trento in Italy. She did her undergraduate education and PhD from the University of Udine in Italy and was a research fellow at Lund before joining Delft as an assistant professor in 2017. Among other awards, she received the CI PhD award in 2016. Her research interests include the analysis and control of dynamical networks with applications to biology and epidemiology, which is the topic of today's lecture. Giulia.
Thanks a lot to Antonis for his very kind introduction, and thanks to all the organizers for inviting me, and good afternoon, everyone. It is a distinct pleasure and an honor to be here with you today and share with you the research I'm passionate about. In particular, I'll be talking about my personal journey across biology and epidemiology, revolving around the astounding robustness of the systems that we encounter in nature. Natural systems across biology, ecology, epidemiology can be seen as nonlinear dynamical networks, namely nonlinear dynamical systems composed of several subsystems with nonlinear local dynamics that are interconnected by an ensemble of local interactions according to a precise topology at various levels of complexity. So the interplay between the local dynamics at the nodes and the local interactions associated with the arcs gives rise to interesting emerging global behaviors. The underlying network structure of these systems captures the interaction topology along with qualitative features and is independent of the specific functional expressions and parameter values associated with the nodes and the arcs. Dynamical systems and control theory, together with graph theory, can capture the underlying structure of the problem, which then we can exploit to analyze the system dynamics and to enforce the desired behaviors. Our journey today will explore two main types of nonlinear dynamical networks from the viewpoint of robustness, dealing with uncertain parameters and keeping the peculiar structure of these systems in mind. On the one hand, we have chemical and biochemical reaction networks. On the other hand, we have epidemiological systems describing the spread of contagion within a population, which can be cast within the same modeling framework and can help us understand and control epidemics. A set of biochemical reactions can be described as a flow graph, where the nodes represent chemical species and the arcs represent reactions that turn some species into others. G functions are monotonically increasing because the reaction rate increases when the concentrations in the starting nodes increase. We can write a dynamical system that describes how the concentrations of the various species evolve over time, and then we can study the dynamic and steady state properties of this system to gain a better understanding of its behavior. We can write the generic biochemical reaction network as a nonlinear flow system where matrix S captures the stoichiometry of the chemical reactions and vector G stacks all the reaction rate functions, and the external influxes are represented by vector G0. Graphical descriptions are precious to capture the system structure, forgetting about the parameters. So matrix S embeds the system structure together with qualitative properties of vector G. And I will show soon approaches that allow us to assess parameter-free properties just based on the structure. And also contagion can be seen as a chemical reaction. Mean field compartmental models for epidemics can be seen as chemical reaction networks with mass action kinetics. So we're dealing with the very same class of models, and we can use the same methodologies to analyze and control them. But why are we particularly interested in the structure of these systems, namely in the graph topology, the wiring among all the various key players in the system? Why are biological networks so remarkably well behaved in spite of their huge uncertainty and their huge complexity? Well, biological systems are often very large and extremely complex webs of myriads of activations, inhibitions, and reactions, but in spite of their complexity, they exhibit an extraordinary robustness that can be found at all scales, from protein interaction to gene regulatory networks, from single cells to whole organisms and species. And in spite of the huge uncertainties, perturbations, parameter variations, and environmental fluctuations, they are able to preserve some fundamental properties and qualitative behaviors that are crucial for survival. Classic examples of biological robustness are the circadian clock, which oscillates at the same frequency across orders of magnitude of variations in the parameters, and the tumbling frequency in bacterial chemotaxis that remains unaffected by huge step perturbations and chemical gradients. So, can we explain the robust behavior of a system exclusively based on its inherent structure, regardless of parameter values? First of all, what is a structure? It is a qualitative description of a whole family of systems, typically based on the underlying graph topology along with qualitative information, and the choice of quantitative parameter values and even precise functional expressions identifies an element in the family. Our structural analysis aims at revealing structural properties that are satisfied by all the systems belonging to this family specified by a structure without resorting to numerical bounds. And these properties are thus exclusively based on the structure, and they're independent of quantitative parameter values, which are often uncertain or unknown or time-varying. So a structural property is even stronger than a robust property. A robust property is preserved for large parameter variations within a set. A structural property is parameter-free. It holds for all possible, physically meaningful values of the parameters. An example of structural property is given by the role of signed loops in systems with a sign-definite Jacobian matrix. Negative loops are associated with stability or with persistent oscillations, and positive loops with the appearance of multiple stable equilibria. In biology, oscillations are important within cells and also in heartbeat, sleep phases, circadian rhythms, and so on, while multistability is crucial in cell differentiation and in ruling cell fate, life cycle, cell division, apoptosis. And we can associate a system having a sign-definite Jacobian with a graph whose nodes represent state variables and whose arcs represent the non-zero Jacobian entries, each with its positive or negative sign. Then we can look at the sign of all the cycles that exist in the Jacobian graph without having to know the exact values quantifying the strength of these interactions, because we are adopting a structural viewpoint. So if all the cycles in the system are negative, we have a structural candidate oscillator. If the system is destabilized, it will necessarily yield persistent oscillations associated with the limit cycle. Instead, if all the cycles are positive, we have a candidate multi-stable system. Destabilizing the equilibrium necessarily leads to the appearance of additional equilibria and multi-stability. This classification has allowed us to gain insight into some physiological mechanisms. For instance, in a system that represents regulation of blood pressure, there are two structural oscillators that are concurrently present, one acting through blood volume regulation and the other through vasoconstriction. And the onset of Mulder-de-Bachmann syndrome that leads to a permanent feeling of movement after a boat or a plane trip may be explained through the internal modal principle and the presence of a structural oscillator that is initially activated to cancel the external oscillation and then becomes permanently active even after the external oscillations have ceased, leading to the disease. Also, the onset of diseases such as fibromyalgia and amyotrophic lateral sclerosis can be explained by the presence of a structural bistable mechanism due to a positive loop and a bifurcation that triggers the transition from the healthy equilibrium to the disease equilibrium. The structural classification can be exploited also to design systems that are guaranteed to exhibit the desired behavior, even in highly uncertain environments. In synthetic biology, we can design a biomolecular clock that is guaranteed to oscillate thanks to the negative loop structure. or we can design a biomolecular switch that is guaranteed to exhibit base stability thanks to the positive loop structure. But in general, assessing structural properties that hold for a whole family of systems is very challenging. How can we decouple the structure and the parameters of a system so as to perform its structural analysis? Well, the tool we have proposed is the BDC decomposition. For a nonlinear system with a stoichiometric matrix S and a vector G of monotonic functions, the Jacobian matrix can be decomposed as a product of three matrices, where B and C can be immediately built based on knowledge of the network topology, of the graph structure, and the diagonal delta matrix in between carries on the diagonal the absolute values of all the non-zero partial derivatives in the system. So this is the local BDC decomposition, which is unique up to permutations. Essentially, any nonlinear system whose Jacobian matrix is the positive linear combination of rank 1 matrices admits a BDC decomposition. And this is practically always the case in biochemical reaction networks, but also in most flow networks in general, not just in the life sciences, but also in engineering. And then an integral formula allows us to bring the BDC decomposition from a local to a global level. So given an equilibrium x bar for the system and the shifted variable z, the global BDC decomposition allows us to rewrite exactly the original nonlinear system in the shifted variable without any type of approximation. And then the constant matrices B and C are the very same matrices in the local and in the global decompositions, and they capture the system structure, the wiring of the key players. The unknown parameter values are isolated in the positive definite diagonal matrix, well, delta in the local case, D in the global case, and we don't care about the precise values or even the functional expressions in delta and in D, because when we are assessing a structural property, we look for properties that hold for the whole family of systems that have the same matrices B and C, with any arbitrary positive values on the diagonal of matrices delta and D. What can we assess based on the BDC decomposition? Well, for instance, we can check whether a family of systems is structurally stable for all possible parameter values. Classic results to assess stability of chemical reaction networks, regardless of parameters, only based on the network structure, rely on mass action kinetics, typically. So mass action kinetics means that there is a specific polynomial functional form for the reaction rate functions. And a classic example is the zero deficiency theorem by Feinberg. We have proposed a new class of polyhedral Lyapunov functions that allows us to generalize structural stability analysis to the case of generic monotonic reaction rate functions. And also the dual class of piecewise linear in rate function has been proposed by Al-Radawi, Angeli, and Sontag. In particular, based on the BDC decomposition, we can guarantee structural parameter-free stability by finding a piecewise linear or polyhedral Lyapunov function. So given the graph structure, we can build matrices B and C of the BDC decomposition, which can then be used in an algorithm that generates the unit ball of the polyhedral Lyapunov function, well, if any, that guarantees the stability of the whole family of systems, which is then guaranteed to be structurally stable, regardless of the parameters, for all possible choices of the positive diagonal entries of matrix D. And to ensure asymptotic and global structural stability, the key is assessing the structural known singularity of BDC for all possible choices of the positive definite diagonal matrix D. And this can be checked through a vertex algorithm. So if the system admits a polyhedral Lyapunov function, then there is a locally asymptotically stable equilibrium, if and only if BDC is structurally non-singular. And if the procedure generates a polyhedral Lyapunov function, also global asymptotic stability and convergence are ensured if and only if BDC is structurally non-singular. But why are we particularly interested in polyhedral Lyapunov functions? Well, because even in very simple examples like this one, it can be shown that the only possible structural Lyapunov function is indeed polyhedral. Other types of Lyapunov functions, such as quadratic Lyapunov functions, could certify the stability of individual systems with known parameters, but they would depend on the rates. So polyhedral Lyapunov functions are key to enable a rate-independent, parameter-free approach in general. Also, as I mentioned, piecewise linear in rate, Lyapunov functions can be seen as the dual class of functions. And indeed, there is an interesting insight that comes from duality. So given a chemical reaction network with stoichiometric matrix S, the dual system is characterized by the stoichiometric matrix S transpose. And the two systems here have different size in general, so their Jacobian matrices are not the transpose of each other. It's not a trivial case. And then, given these two dual systems, within the stoichiometric compatibility class, possibly induced by the presence of conservation laws, so conservation of mass, the primal system is structurally locally stable if and only if the dual system is. In both cases, we can also consider the systems where the state variables are no longer the concentrations of the chemical species, so X and Z, but are now the reaction rates R, which is G of X, and W, which is H of Z. And also these systems in R and in W are the dual of each other. So we can write the four systems, primal and dual, in concentrations and in rates, according to the global BDC decomposition, and then we can adopt the BDC-based algorithm to compute a structural polyhedral Lyapunov function, if any, for each of these systems. And we see a nice and interesting symmetry in the architecture of these systems, also in relation to global stability assessed through Lyapunov functions. For the two pairs of systems, so this one in yellow and this pair in blue, one system admits a polyhedral Lyapunov function if and only if the other system admits a polyhedral Lyapunov function, which turns out to be the dual polyhedral norm. So if one wonders which type of function is the most useful, like piecewise linear or piecewise linear in rates, well, they're both equally useful, and they are essentially indeed the dual of each other. They're two faces of the same coin. And this duality can be precious to assess the structural stability of particularly challenging architectures, of particularly challenging structures, through the analysis of their dual structure. The BDC decomposition can also be used for robustness analysis when parameter values are unknown, but now bounded within known intervals. And then we can derive some vertex results exploiting convexity and multi-affinity that allow us to assess whether a property holds in a whole box in the parameter space by just checking whether the property holds on the vertices. For instance, to perform robust stability analysis, the BDC decomposition allows us to rely on sufficient conditions based on the well-known mapping theorem and zero exclusion theorem, so relying on fundamental classic results in the systems and control field. And in the presence of a constant input perturbation affecting the system, the BDC decomposition allows us to leverage vertex results to determine robust lower and upper bounds for the input to output sensitivity, exploiting the fact that, again, it is a multi-affine function of the uncertain parameters in the diagonal matrix D. And similarly, we can obtain robust lower and upper bounds for the magnitude and phase of the transfer function when the system is affected by periodic input perturbations. So what else can the BDC decomposition tell us? The BDC decomposition can also help us structurally predict the sign of input-output influences at steady state for all possible choices of the parameters. So we consider a generic nonlinear system resting at a stable equilibrium point. We apply an input perturbation, not so strong to destroy the system properties, and we look at the evolution of a quantity of interest labeled as an output. So after a transient, the output will land on a new equilibrium. Is this new equilibrium value always larger than it was before, regardless of parameter values? Then we have a structurally positive influence. Is it always smaller than it was before, regardless of parameter values? Then we have a structurally negative influence. Does it always go back to the very same value it had before, regardless of parameter values? Then we structurally have the so-called perfect adaptation. But if the behavior depends on the parameter values, then there is no structural influence. So we have a question mark. So we can look at the structural steady-state influence of variable j on variable I, which is the sign of the steady-state variation of xi due to a step input acting on xj. So it can be plus, minus, 0, or it can be a question mark when it depends on the parameters. Mind that this influence is not just the direct influence associated with entry ij of the Jacobian matrix. We're considering a class of systems that is much more general than systems having a signed Jacobian matrix here. So this is the net influence, which takes into account both direct and indirect influences coming through the network topology. And exploiting the BDC decomposition allows us to evaluate structural influences through a vertex algorithm, once again exploiting multi-affinity. So if the influence has the same sign at all the vertices of the parameter box, then it has the same sign for all the choices of the parameters within a continuum space. And the BDC decomposition also allows us to compute the influence matrix, which considers all the possible input-to-output pairs for a given system with both input and output associated with one of the state variables. This allows us to assess interesting features of real biological systems, such as this osmoregulation system in the bacterium Escherichia coli, where almost all the influences, as we can see here, are structurally signed. So they're completely unaffected by parameter variations because they just depend on the system architecture, the system structure. And if we compare the structural influence matrix with experimental results, then we have a precious tool for model falsification. So for instance, here we see that if we apply a step perturbation to species B, then the new steady state of species A is going to be always larger than before. We have a structural plus here. But if we happen to observe experimentally that the new value of species A is smaller than before in even just one of our experiments, then we can conclude that this model is not suitable to represent the phenomenon that we're looking at, because it will never be able to reproduce the behavior that we have just observed in the lab, no matter how we pick the parameters. And an interesting case study here is given by the mechanism of protein-based intracellular ceramide transfer from the endoplasmatic reticulum to the trans-Golgi network. And for this phenomenon, two alternative models have been proposed in the literature. One is the short-distance shuttle model, where the protein, SERT, goes back and forth between the two membranes to deliver ceramide, like a ferry, like a shuttle, indeed. The other mechanism is the neck swinging model, where this protein SERT needs to be attached to both membranes at the same time in order to deliver ceramide by bending its neck back and forth. So this is the neck swinging model. So can we structurally compare these two alternative mechanisms that have been proposed by biologists to explain the very same phenomenon? So we can see both models has dynamical systems where the flows between compartments are driven by the amount of other species in the system. And then the two different transfer mechanisms are associated with two different topologies of the associated dynamical systems. And using the BDC decomposition, we can compute the structural influence matrices of the two systems. And from these, we see that the two models are not structurally incompatible. So the influences either have the same sign in both cases, or one is signed and the other one is a question mark, or they're both question marks. Still, while the influence matrix allow us to discriminate between the two models if we design suitable experiments. For instance, if we see in some experiment that we have a positive influence corresponding to entry 1,3 here, then, well, the first model has a question mark, so it can still be valid, but the second model here has a minus, so it would get falsified by this experimental outcome. And structural analysis also reveals that the system is structurally tailored so that the concentration of phosphorylated PKD here, seen as an input, promotes a special flow. So we have a structural flow regulation in both cases, but in the two cases we have a different flow that gets regulated. In the short distance shuttle model, this flow is the flow that moves the protein set back and forth. So between the two membranes. So the transfer of ceramide associated with this movement back and forth gets structurally regulated. In the neck swinging model, instead, the flow that gets structurally regulated is the flow that detaches and attaches SERT to both membranes. And this structurally favors the mobility of SERT in the cytosol, preventing it from remaining always attached in the same area, even after that area has been already depleted of ceramide, so there is nothing more to be transferred. So, which is the right model? Structural analysis is powerful. It captures the essential structural differences, indeed, between the two models. However, it is not enough for us to fully distinguish among them. For this, we would also need to consider some parameter-dependent properties, most likely. And indeed, in general, structural analysis offers strong insight into parameter-free properties of systems. But when a property is, in fact, parameter-dependent, which system features can enable that property or prevent it? Which key parameters need to be finely tuned in order to enforce the property? Well, structural approaches cannot answer these questions. On the other hand, well, numerical simulations at the other extreme will only focus on single snapshots of the parameter landscape, and they cannot reveal the essence of a phenomenon, of a mechanism. So to gain a deeper insight into natural phenomena, we need to bridge this gap between structural, so unknown parameters, completely parameter-free, and repeated numerical simulations approach. Because indeed, well, both structural and robust analysis have a yes-no attitude. So we're wondering whether a property holds regardless of parameters or for all parameter choices within given bounds. And when the answer is no, we're left clueless. We can only numerically simulate the system case by case. Well, to obtain both qualitative and quantitative insight, it would be great to be able to go beyond structural approaches and integrate structural and robust approaches with probabilistic and randomized methods, so as to check whether the property is structural or robust or holds with guaranteed probability, and identify the crucial components or motifs or portions of the parameter space or parameter values or relations among the parameters that are fundamental to enable or to prevent the property. We have preliminarily explored a probabilistic analysis approach in the case of opinion formation models to assess the probability of some qualitative outcomes such as perfect consensus, consensus, polarization, clustering, dissensus, when we know the type of model, so the model structure and the type of network, but we have no information about agent parameters and initial opinions. We only know intervals and distributions. Also, we have introduced recently a definition of resilience for a family of ordinary differential equation systems consisting of stochastic perturbations of a nominal deterministic system. So we have a deterministic nominal system that is known to have an attractor, A, and a basin of attraction, B of A. And then all the other systems in the family are obtained by adding a stochastic noise on top of these nominal deterministic dynamics. And then if the family of systems is we consider this family of systems, and we say that it is practically resilient if for each element within the family, in spite of the noise, for all the initial conditions inside the basin of attraction and at an epsilon distance from the attractor, the system trajectory remains within a delta distance from the attractor for the whole time horizon from 0 to tau, with probability at least gamma. And we can also consider an asymptotic concept of resilience and essentially require that all the trajectories emanating from the basin of attraction will asymptotically eventually converge to a delta neighborhood of the attractor with probability at least gamma. And then we can also consider the attraction time, tau, which is such that the probability of the state to return to a knee neighborhood of the attractor after time tau is at least me. And we have applied our definition to study the resilience of, for instance, Turing pattern formation in a population model, phytoplankton and herbivores. The probability that the system trajectories remain within a delta distance from this oscillatory attractor increases with delta, as expected, and also decreases for increasing magnitude of the noise, lambda. And conversely, the attraction time becomes smaller for increasing delta and becomes larger for increasing lambda. And also for the case study of stability in gene regulation models, so in this gene regulation model where depending on the parameters we can have 1 or 2 stable equilibria, we can look at the probability of remaining within a delta neighborhood of this equilibrium here. This probability, again, as we can see from here, is larger if delta is larger and if the noise intensity lambda is smaller. And the attraction time, on the contrary, is smaller if the probability is larger. I mean, we can also consider how resilience is affected by the distance of the system parameters, and in particular of the system parameter A, from its bifurcation value, which is the bifurcation value that makes the considered equilibrium x3 vanish. So the perturbed system trajectories remain within a neighborhood of the equilibrium when the bifurcation parameter is sufficiently far from its bifurcation value for all simulated noise intensities. However, as the parameter tends to its bifurcation value, then the probability associated with practical resilience decreases sharply. So far we have focused on biochemical systems, but as we have mentioned, also contagion can be seen as a chemical reaction. So the same modeling class embraces compartmental models for epidemics of arbitrary complexity, including, for instance, the Siddhartha model that we have proposed to capture the peculiarities of the recent COVID-19 pandemic. And that includes a bunch of different infected classes, depending on whether the infection has been detected or not, and the severity of symptoms, asymptomatic, symptomatic, acutely symptomatic. And this model emphasizes our control knobs to enforce physical distancing and lockdown, acting on the contagion parameters, and also testing and contact tracing, acting on the diagnosis parameters. And another control action is related to vaccination, which can be considered. And more in general, we can consider epidemiological models with an arbitrary number of infected categories. For instance, symptomatic and asymptomatic, detected and undetected, and so on. And here vector X stacks all the possible infected categories, so the fractions of populations in all the various infected categories, while S is the fraction of susceptible population. The system depends on uncertain and time-varying parameters, these like P and V. And this makes the epidemic evolution particularly challenging to predict. We have uncertain flow parameters that describe how individuals move between different infected categories. And we have infection parameters that describe the contagion occurring when susceptible individuals meet infected individuals in any of the infected categories. And we assume that these parameters are bounded within known intervals. And we can see the whole system as the feedback of a positive linear system describing the time evolution of the infected categories, fed back by the system describing the time evolution of susceptible people. And in fact, we can leverage positivity of these vectors here, and the fact that this matrix f of p is a Metzler matrix, which is also Hurwitz for all possible parameter values. And so f can be written as the positive linear combination of rank 1 matrices, so it admits a BDC decomposition. And thanks to this, we can have interesting vertex results concerning the well-known reproduction number, R0, whose value determines the stability of the disease-free equilibrium. So for parameters P and V varying in a box, R0 achieves its minimum and maximum values on the vertices of the box, again, in light of multi-affinity. And we can also provide lower and upper bounds for the epidemic evolution using optimal control approaches. So our cost function includes both a terminal cost and an integral cost to be able to capture, for instance, either the total number of infected or hospitalized individuals at the end of a finite horizon, or the cumulative number of infections, hospitalizations, and deaths. And then we can solve the optimal control problem by considering the uncertain parameters as controls, and S as an uncertain parameter, an uncertain quantity that is bounded within an interval. This is justifiable since S varies slowly, and in some cases it can be even controlled by us through vaccination. And then we can compute the optimal control with a single backward integration without having to resort to shooting, thanks to the positivity and to the strong structure of the system. And for instance, for the Siddhartha model, here are some guaranteed bounds for the evolution of the fraction of infected people and of people in intensive care units. So upper bounds are in red and lower bounds in blue. And in black, we see system trajectories with parameters that are randomly generated within their intervals. So optimal control problems can be solved to identify the worst case and best case scenarios, worst case and best case values of the parameters, which helps us indeed make predictions if we cannot change these parameters, and also helps us plan interventions to drive the parameters to their best case value if we can affect them, if we can change them. And we can generalize epidemiological compartmental models even further by considering not only an arbitrary number of infected categories, but also an arbitrary number of non-infected categories, for instance, susceptible, vaccinated, recovered. And then the uncertain parameters describe, well, the flow among infected compartments along with recovery in F, vaccination and waning immunity that drive the flow among non-infected compartments in G, contagion parameters driving infections in C, and recovery parameters in D. And we can write in this form, for instance, an extended Siddhartha model that includes not only vaccination, but also waning immunity and possibility of reinfection for both vaccinated and recovered. And for systems of this broad class, we can formulate an optimal control problem to design optimal vaccination strategies. And we have proven that this sequential quadratic Hamiltonian algorithm for optimal control, which had never been used before for epidemiological models, can actually solve the problem efficiently and has proven rigorous guarantees for global convergence. And it can also handle non-smooth costs and allow us, for instance, to track a desired discontinuous vaccination profile arbitrarily well if the weight on this non-smooth term in the cost is sufficiently large. So I'm now concluding my overview of this personal journey through systems in nature from biology and epidemiology, guided by their structure and their outstanding robustness properties in spite of uncertain and time-varying parameters. We have seen how mathematical approaches building upon the BDC decomposition and systems and control methodologies and graph theoretic methodologies can give us insight into structural behaviors and allow us to perform structural analysis and robustness analysis to explain robust properties, obtain robust predictions of the evolution of uncertain phenomena, and devise robust and optimal control approaches. And we have seen that we can, and we would like to go beyond and integrate structural and probabilistic approaches to gain a deeper qualitative and quantitative insight and understanding of these systems. And there are a lot of interesting open challenges to be tackled in this direction. Let me thank all the friends and colleagues and group members with whom I've worked on these topics. Thanks a lot again to the organizers for inviting me, and thank you very much for your attention.
Thanks, Giulia. I think we have time for a couple of questions. Yeah. I think there's someone coming down. With the right microphone.
Okay. Can you hear me?
Yes.
Okay. Thanks for the presentation. In the beginning of the talk, you talked about stability in the sense of a structural perspective, structural approach to stability. When you talked about robustness, the approach changes to parameter variation. Do we have some result about the robustness of the system from a structural approach, meaning that the system is structurally robust? In the sense, for example, the system is robust despite deletion or addition of links.
Okay, yeah, that's indeed, that's a slightly different flavor of problem. There are results from the complex networks community that are very much focused on checking whether the alterations to the graph topology would lead to a different behavior. They're mainly focused on static behaviors, so joining forces with the dynamical systems and control community would be beneficial to try and understand how dynamic behaviors are affected by changes in topology. Something has been done in this regard. What I was focusing on was indeed, like, by structure, I don't mean related to alterations or not of the topology, but I mean indeed for all possible parameter values given that the topology is fixed. But there is ongoing work on indeed also related to topology dependent or topology independent results. Actually, I've recently worked on, essentially, you can prove that if you have a biological network where all the interactions are of the Michaelis-Menten type, you have guaranteed stability under some technical assumptions, regardless of the topology. So no matter how the components are, so it's not just parameter-free, so it does not depend on the parameters as long as the type of functional relation is of the Michaelis-Menten type, but you have stability also regardless of the actual wiring among the components. So indeed, there is also ongoing work in this regard. Okay, thank you.
Thank you.
Thank you.
Thank you.